Alright, I'm doing this series because I think it can be helpful to have different explanations of stuff. So BUGGED episode 3, we're talking about humoral immunity. This is a continuation of last time. Essentially, we're talking about the different types of antibody response. Okay, so last time we left off by talking about the heavy chain and the light chain, and the light chain and the heavy chain both have variable regions. Those are determined by loops of protein called the complementarity determining regions, CDL1, 2, and 3, and those in turn are coded for by BTJ segments. Your body is going to choose to marry different V's, different D's, and different J segments together to make a unique protein output. So remember, it's like kind of like a menu, choosing a starter, one of a dessert, and one of a juice menu to kind of form a unique combination. And in the light chain, remember, they do not have a D region. Okay, terminology, this is the FAB, the antigen binding site, and this bit here is the FC region, the constant kind of region. So remember, when once the VDJ genetic kind of bit is combined, it's fixed for life, okay? I kind of think of it as like a face. So everyone has a different face, and that's your different variable region, and your face is going to kind of stay the same for life. However, obviously, every day you can wear a different outfit and kind of look different still. So depending on your drip, you can look different, and your drip essentially is your constant region. So although your face is the same, i.e. your variable region is the same, your drip can be different because you can change your constant region. Essentially, that is known as isotype switching, which we'll be talking about today. Okay, so imagine you've chosen a unique V, D, and J sample. So one of each, like choosing a pack of cards from a menu, for example. For example, number 34 from the V sections, number 18 from your D options, and number 4 from your J options. And those are now glued together, and that is permanent genetic recombination. So all your cells in the future are going to have V, D, J kind of segments. That's kind of like your face. So your face is now set in time. However, later on, mutations can slightly alter these genes very slightly, but we'll be talking about that later on. But essentially, once the genetics are set, either through this or later on through mutations, they're not going to be changed ever. However, you can change your drip basically on a daily basis. So depending on where you are in the body, you can have a different constant region, which you will choose not by permanent recombination, but you'll choose it instead by temporary splicing. So remember, the exons is the coding part and the introns is the non-coding part. So what we'll do, we have 5 options for the constant region, and your body only chooses one of them. So it's going to consider all of the rest of them as introns, and only one of them, for example, the gamma, as an exon, as a coding region, and then join that together with a VDJ to make a VDJ with a constant region gamma. And that is going to give a different outfit than, for example, if an alpha would be combined. So we'll see what I mean here. For example, if we choose the VDJ and then marry that together with the gamma exon and cut out all of these via splicing, then we're going to end up with our most common type of antibody, which is called an IgG, and that is our main circulating antibody. If, for example, we choose, instead of the gamma, which is an epsilon or a delta, we are going to end up with an antibody, and it's going to have the same variable region, remember, so it's going to always have the same face. However, the alpha is going to be different. So this is going to, for example, if you choose the epsilon one, it will be an IgE kind of outfit, and if you choose a delta one, it will be an IgD, so slightly different kind of color maybe, for example, with slightly different color trousers, but it will still be in the same shape. So IgE is the main antibody responsible for allergies, and IgD is, remember, on your B cell. The B cell has a receptor called the antibody, and the antibody on your B cell is called your IgD. And these guys here, this is secreted by your plasma cell. This IgG is on the surface of your B cell. Okay, say you choose an A combination. So this is, by the way, E and D will give slightly different variations, but the point is they're the same shape. However, if you choose an A segment as your exon and cut out all the rest as introns, and then join your A to your VTJ, so remember your VTJ is the same, you're going to get some kind of techie shape which is like a divalent antibody. It's going to basically get one antibody and then the same antibody symmetrically, and then join them together with a joining segment, not this J segment, but a joining segment here, and make a divalent kind of dimer. This makes IgA, which obviously you can tell is different from all of these guys. And this is the type of antibody found at the mucosal surfaces with a joining segment. So the mucosal surfaces are, for example, called the MALT, the mucosal associated lymphoid or the GALT, the gut associated lymphoid. So these guys are secreted at the mucosal surfaces. However, note again, the variable region is the same for all of them. So all of these will be able to bind to exactly the same antigen. However, the way they're secreted is essentially different. The way the kind of shape is different, but the shape of the variable region is the same. Okay, and finally, if we use our mu constant region and splice out all the others and then marry this with our VDJ permanent gene, then if you thought this was techie, this is even more crazy. We get a pentavalent antibody thingy. So we have 5 antibodies joined together, joined by disulfide bonds. Remember, okay, with the antibody, this is one binding site, and this is another binding, for an antigen, this is one binding site, and this is another binding site. So each one has 2, and if there's 5 antibodies joined together, 2 times 5 is 10, so there's 10 potential antigen binding sites. Okay, now we're going to take a break to talk about fishing. So I'm not sure if any of you do fishing, but I've thought that there's 2 ways to do fishing. from my limited research. So you have like a really exotic fish, like a rare fish. You can either spend time studying what does this fish exactly like to eat, what attracts it, etc. And you can make a bait on a fishing rod that is tailored to its taste, and hopefully a fish will come and bite. So your fish is your antigen. Or you can go crazy and just be like, I don't have time for this, and just put out a massive net in the middle of the ocean, and eventually your fish is probably has a likelihood of swimming into it anyway. So these are two methods. And essentially this corresponds to two types of antibodies. The studying the fish for a while to find out the best and the tastiest bait is your IgG antibody. Essentially, it's going to adapt the variable region ever so slightly and spend time doing that so that it can be tailor-made for that antigen and be ultra specific. However, your IgM immunity is like, it doesn't have time for that. Basically, this is the net strategy. All it does is it's like, yo, I'm going to have loads and loads and loads of binding sites. It doesn't matter if they're less specific, because there's so many, there's a high chance that the antigen is going to fit into one of these many surfaces anyway. And because basically studying the bait, essentially studying what the fish exactly likes, that takes a longer time. So because basically it takes a longer time, and basically you have to perfect it. However, if you just throw a net in the middle of the ocean, that can be done fairly quickly. Obviously you have to know where to put your net, so you have to have a rough idea of your variable region, but it doesn't have to be as specific as your IgG. So this is where the concepts of affinity and avidity come in. Affinity is this idea of how specific is the antigen to one specific binding site. So for example here, how tight is that bond? Avidity, on the other hand, is your overall kind of binding strength. So although the antigen may bind kind of more weakly overall, because it's to one specific site, overall because there's more, there's just so many binding sites, there's a high likelihood of the antigen kind of fitting in. And this transition kind of happens. We'll talk about how this transition happens. So this leads us to this graph, which is essentially your immunity graph, your immune memory graph, or your vaccine kind of graph. So you have exposure to an antigen first, and then a few weeks later you have exposure to the antigen again. So we're going to talk about the response that happens. Firstly, we're going to see a peak in IgM, and secondly, after our second antigen injection, or after a while, then we see the IgM peak go down, and after a few weeks the IgG peak goes up. So essentially, remember the IgM is like a net, essentially. It's not as strongly able to bind to stuff, and we'll see why. But because it's so huge, it has a higher chance of essentially catching the antigen. And later on, essentially, we're going to get your body has time to kind of study the antigen, study the bait, study what kind of bait is needed, and then make a very specific kind of fishing rod, and that comes through your IgG. So this transition from your IgM to your IgG essentially occurs because of mutations that occur in the VDJ segments that make the antibody even more specific. So essentially your body takes time to study what makes that antigen really, really tightly bind to the antibody. And this is in a process called somatic cell hypermutation. So this occurs after antigen recognition. Essentially this, we'll talk about it in another video. Essentially, it takes time for your body to work out how can we optimize this antibody to be absolutely amazing. And that is your fishing rod response. But initially, it doesn't know what it's doing, basically. It kind of has a rough idea, but it wants to have the highest chance of catching the antibody as possible, so it puts the huge net out. However, after a few weeks, it doesn't need the net anymore, so the levels of IgM decrease, and the levels of IgG increase. And then upon second exposure, your body kind of knows all it needs to do to make this IgG. So on second exposure, it's very, very happy to make this very, very quickly, and it doesn't need to make as much IgM. Obviously, you will have some, but it doesn't need to make as much. Hopefully that kind of makes sense. The other reason why IgM is going to decrease after a couple of weeks is because this kind of structure, essentially the structure of IgM, they can put this, for example, each antibody can bind to an antigen, and then that antigen can bind to another antibody, which is on another IgM, and that can bind to another antibody. And essentially what you can have is these IgMs, because they're kind of like big nets, the nets can kind of get entangled with each other, and that is called, that basically is called forming an immune complex. These immune complexes are insoluble, and they can end up in tissue, and therefore cause local inflammation, and this is known as type 3 hypersensitivity. So in order to avoid this kind of immune complex formation, your body limits the amounts of IgM that it puts out, and as soon as IgG is ready, the levels of IgM decrease. Again, if you're not sure about what I'm talking about here in somatic cell hypermutation, the next video is on that. Okay, just briefly, what do the different IgMs, what do IgM and IgG do? And how do they actually kill bacteria? So IgM works, remember IgM is our pentavalent one, our net. This comes out first in your immune response. And this works in only one method. It works by activating your complement system via the classical pathway. The classical pathway is antibody, and then goes QRS, and then activates complement. C3 protein is converted by C3 convertase, and then we get obviously the C3, A and B, and then opsonization, phagosynthesis, and all that. The first video is on the complement. How does IgG work? So IgG, remember, is the main circulating antibody. However, it takes more time to come about because it has a essentially low avidity but high affinity. So essentially what that means is it takes your body time to figure out how can we tweak this to be super specific to the antigen. It works in two ways. So firstly, this kind of works by binding to toxins and can neutralizing them. So if this binds to a toxin, it stops the toxin from binding to the cell. Basically, it kind of swarms the toxin, stops it from binding to the cell, and therefore stops a toxin entering your body. However, remember, IgG takes time to be made in the first place, and it also takes time to be released. So in order to be effective as an antitoxin, it should be circulating in the bloodstream in the first place. So this is why, for example, if you've had a toxin, you can't wait for your body to make the immune response. You'll be given an antitoxin, which is essentially you'll be given passive immunity. You'll be given IgGs that have already been made, like in a mouse or in another human. You'll be given them in your bloodstream, and those will very quickly stop the toxin from working. This is the same kind of competition principle that works with viruses. So it can bind to viruses and stop a virus binding to receptors in your cell surfaces to stop them entering your cell. The other way that IgG works is that it doesn't activate complement, but what it can do is it can directly coat bacteria and then tell macrophages and neutrophils to munch them, essentially. So this is your antigen binding region, your FAB region. Your antigen binds to this bit. So let's say the red pointer is your antigen. This the antibody basically acts as a flag and sounds the alarm bells, and then tells macrophages, which wouldn't be able to detect this tiny antigen on its first place. However, with the antigen bind to it, the antibody kind of sends off alarms and tells the macrophage to bind to the FC region of the antibody. Remember, the FC region is this kind of straight bit, and then from there it kind of munches, it goes upwards and munches your antigen. So that is how IgG works. Okay, finally, here's some questions to do.